So the last 2 hours, and the last hour was chaotic and people liked it. So I want to keep it chaotic this last 2 hours as well. I hope you will enjoy it. Do you remember where we were last time? What did we see? It was chaotic, right? So let me just go randomly. Like there's some literature here. I can talk about that. Codes, spirals. Let me talk about spirals. So I already mentioned reaction diffusion to you briefly, right? Let me mention it again. So this is one of those triumphs of exploration in computational universe, because it's back in 1950s. DNA was found in 55, 60? 55 or 57? Who knows? Yes, thank you. Well, we don't know much about biology, but Turing is interested in how these patterns appear on animals, right? So he proposes a mechanism for it. It is called reaction diffusion. Basically, imagine our systems with limit cycles. Okay, imagine you have a nonlinear system and it creates, let's say you have a nonlinear system. F U V and G U V, and it has a limit cycle. Okay, 56, 53. So it's before that. Okay, U V. So let's say it has a limit cycle. Everything approaches to this, and whether you start from a different condition, you approach a limit cycle. What happens? So this thus far, it's just U and V. So there's a system, and the system is defined by sort of a lumped parameter, U and V. But what if it's a distribution? What if there is a U of xy and V of xy? It's distributed, and you put a diffusion here. Diffusion was something like this, right? So while you have a limit cycle, you also diffuse. What will happen? Well, what happens is that you get these spirals, those waves that we have seen in Belousov and Jabotinsky. So you can think of this U and V as chemicals, maybe. They interact and they form an autocatalytic set. And as they diffuse in one place, it's an oscillatory chemical reaction, right? While in here, maybe it's in the zero phase. Somewhere here, it's in the pi over 4 phase. Am I making sense? So you create waves that also go to other places. So let me show you a nice example of this. Let's go to literature. Rock, paper, scissors. Mobility. This is a Nature paper. Mobility promotes and jeopardizes biodiversity in rock, paper, scissors games. What is rock, paper, scissors games? So you basically choose 3 bacteria. What happened?
I was going to do this article.
Yes, you can do it still. Yes, very good. So how rock, paper, scissors work? A wins over B, B wins over C, C wins over A, right? So if you put it in in a differential equation, you can imagine that, and if you put some diffusion in it, you can imagine that it will form spirals like so. Like this kinds of spirals. A is eating B, B is eating C, and it rotates like this because in the time that the B eats C and comes and expands over there, C eats A and expands to the other side, so it starts rotating, right? I happen to have a code for it, which is here. No, this is the genetic code. Let me go to Dropbox, teaching, shared, chapters, Darwin, codes, spirals. Yes. Yeah, these are those three species, and this is the diffusion part. And this is where they eat each other. Okay, if you run this code, it starts like this, but then it settles into these spirals. Do you see the spirals? Yeah. Very nice. So it turns out that this is not just something that happens over here and we never see it again. That never happened in this course, right? Something is somewhere and you think that it's arbitrary, then it turns out it's part of the universality stuff, right? So let's put a pin on this kinds of rotating spiral waves. Actually, no, let's not put a pin on this. Let's go to a paper. So I will tell you now, chaotic. I will tell you now a story of a paper, but it happens in stages. So we have a bit lost it. Like when we do, we always think that it has to have a purpose and things like that. But let me start from here. Just this. Once upon a time in a good Moscow day in Pushkina, Pertsov, Yermakova, and Pamphilov thought that it's a good idea to think about rotating spiral waves in a modified Fitzhugh-Nagumo model. Okay, and they come up with this rotating spiral wave. Okay, what is the year? It's 1984. Okay, who is Fitzhugh-Nagumo? Let's also go there. Sorry. Fitzhugh describes a prototype of an excitable system, example, a neuron. We will come to neurons as well, by the way. So it's just a type of an equation that you have already seen, like, right? Like a two-dimensional equation. It's even if you take A and B to be zero, it's exactly what we have seen before, van der Pol oscillator. So it's a generalized van der Pol oscillator. So far, are you still with me? So this Pamphilov changes it a little bit, maybe adds some diffusion, whatnot, and they get the rotating spiral. That's the beginning of the story. We continue with this story, and we come to 1992. What these people want to do is simulation of biological cell sorting using a two-dimensional extended Potts model. So what is Potts model? It's Ising model, but not with just up spin and down spin. It's n spins, okay? And they interact. And they basically write a Hamiltonian. By Hamiltonian, I mean so it's still a cellular automaton. So there are like parts of the system that describes one cell, and then there is a next cell here. Whether this cell will diffuse towards there or not is controlled with this Hamiltonian. What do we mean? Cells want to keep the same size, okay, not grow too much or not shrink down to zero. So that is controlled by this parameter. Do you see? There is minus a squared. So if the area increases too much or decreases too much, it's too high energy. So it's like the stiffness of the cell wall, right? There is also in this particular system two types of cells, and they want to have boundary with each other, the same kind of boundary, and not be together with the other kind of cell. So what happens in the end is that if you run this and everything in here you have learned. You know how to do Monte Carlo stuff, and they are doing Monte Carlo here, probably. How else can they do it? Monte Carlo simulation, I'm presuming. Let's see, MCS, whatever. Do you see what happens? They start with a soup of cells and run this algorithm that has no mind in itself, right? But these cells organize into a region with black cells and white cells. So far, so good? Good. Now comes the third paper. Pamphilov is here now, back, 1999. What they do is they investigate the migration and thermotaxis of Dictyostelium discoideum slugs. Okay? So these are amoeba. Let me just show you their videos first. Tell me, you have a question? No. Let's see if this will be too much sound or not.
Of the cellular slime mold, Dictyostelium.
Are we happy with this? Yes?
It's plentiful. The mixamoeba are completely independent as they feed and multiply. When their food supply dwindles, their behavior changes remarkably. When they become crowded, they start moving together from all directions, flowing into growing masses containing thousands of cells.
Did you see the spirals?
Yeah.
They are chemically interacting with each other. That's the spiral of Pamphilov. Okay? Oh. Why we don't have sound anymore?
Into growing masses containing thousands of cells.
We don't have sound here at all. Okay. It's like Terminator 2, right? Have you watched it? Are you too young to not watch Terminator?
Aggregation starts when one cell spontaneously secretes a pulse of a hormone called cyclic AMP.
Cyclic AMP.
Nearby cells respond by moving towards the hormone and by emitting their own pulse. Soon, this response becomes widespread and synchronized, creating waves of accelerated movement passing through the migrating cells.
It's all related with the course, right? It's not unrelated. You will see anyways.
These mixamoeba are streaming steadily towards a distant aggregation center. As always, there are a few stragglers rushing to catch up. The aggregations of cells round up, and then each turns into a slug called a pseudoplasmodium. These slugs are active, searching the terrain for a suitable place to settle and leaving a trail of slime as they go.
So these are not organisms with brains. This is just Terminator 2. It's just, do you see?
Remember that each slug consists of thousands of free-living amoebae, which now have organized themselves into a cohesive unit and which synchronize their activity. During its travels, an individual slug may fuse with other slugs it happens to encounter. Meanwhile, a few individual mixamoebae are still visible, moving around on the surface. After this migratory activity, the slug rounds up again, but this time it flattens onto the surface and then grows a thin stalk with a fruiting body on the tip. Throughout these maneuvers, the mixamoebae remain separate individuals. During this final phase of reproductive activity, the behavior and fate of each cell continues to be subordinate to a self-generated pattern. The mixamoebae in different parts of the pseudoplasmodium differentiate into three main types of cells with different functions. Some cells form the base and others the stalk, while the rest migrate up the stalk and turn into spores in the fruiting body. The mature spores are later released.
You may be thinking that this is impossible to understand, right? But we are, let me show you this paper. Look, remember the slugs? Here they are. They just start with random distribution of cells, cell types. And here we have those two papers together. So the arrangement of the cells is controlled with that Potts model. While they interact with each other, they signal with each other through, like there's also this CAMP, cyclic AMP, in the environment that helps them synchronize their activity. So as a result, they start moving like a slug, okay? But this paper just understands that behavior and ends, right? And you can see that those limit cycles and whatnot are here. Like inside the amoeba, there is this kind of differential equation. Outside, there is this kind of differential equation. As you can see, it's reaction diffusion, right? It's diffusion and reaction parts. Then there is Hamiltonian, right? Very good project that connects everything. And then there is limit cycle inside. Yes? Very good. So now we go to the ultimate paper, which is this one. How amoeboids self-organize into a fruiting body. Okay, they explained the slugs, how on earth it comes and turns into a flower or kind of a thing. Well, just if you build the correct model, you can just explain it with computation. It's like magic, right? And they understand everything, how this happens, and it's still same kind of model. Let me show you some videos from this paper. For example, this one. Are you impressed at all with this? Yes? It's ultimate computational biology, right? Okay, this is one of the videos. Let me show you another one, which hopefully shows the spirals as well. Do you see this spiral going throughout the thing and helping them organize? Do you see? Very nice. Okay. Do you have any questions? No.
Are we going to talk about zebras?
Let's talk about zebras, yes.
The reason why Turing, invented these direction diffusion PDs was basically to explain why zebras have stripes.
Okay. How the zebras have stripes, right? Not why. Why is a different question. Yes. Okay.
The mechanism between the stripe formation in zebras, because the interesting thing is zebras have widely irregular patterns.
Yeah.
And the patterns don't seem to, correspond to their genetics. And they are very asymmetric. And especially back in 50s, the asymmetry in life was, basically asymmetry by organisms was considered very weird because, everything involving the process is symmetric. How can a symmetric process generate asymmetric patterns in the results?
And what is the explanation?
Turing instability.
Okay.
Basically, the reaction diffusion PDs, they are unstable with respect to initial states. But so small perturbations in the initial state can, create. The small perturbations in the initial states or conditions can create widely differing end results. But these end results always, look like the stripe shapes that zebras have, and despite being unstable with respect to initial states, when time goes to infinity, they converge on something stable. Not the stable thing that converge to depend sensitively on the initial states.
Yes. So we have sensitive dependence on initial states coming from the chaos in the system, right?
Yeah.
And nevertheless, there is also order in the system in a sense that ultimately, if you wait for infinite time, they will all be perfect stripes. But you don't have infinite time, so in finite time, they stay as they are. Am I understanding it correctly?
Kinda, but they won't become perfect stripes. Basically, if I give an initial data and wait infinite time, then it's going to converge on something. But what it converges to depends very sensitively on the initial data.
Very good, very good. So why do zebras have stripes? Do you have the answer to that? Camouflage from what? Lions against yellow sand, black and white stripes.
Lions, they don't see colors. When they're camouflaged,
The predators actually don't observe colors.
Well, that was the original idea. Then they realized that it's a bit different. It's a camouflage not with respect to backdrop, but camouflage with respect to the herd. Okay? Here is how it works. So scientists were confused which zebra is which, so they just painted one of them red, just red dot. Okay? Lions immediately ate that one because it helped them concentrate on one target. Otherwise, zebras are camouflaged with respect to the herd, so it's a superorganism now for lions. They can't decide on which one to chase, so they can't chase anyone if it works perfectly. But if one of them just broke a leg, done, because it can't follow and stuff like that, or have different pattern. So they can decide this the weird one. Let's go for the weird one. Same with everything, like fish and everything. They have the same kind of camouflage.
And then it actually keeps that evolution, like only the ones with correct stripes and
Yes.
the ones that are in this
Yes, exactly.
suitable.
Exactly. Very nice.
Is there something like this in giraffes as well?
Giraffes.
Because they also have.
Well, I can manipulate your question to tell something else. Like giraffes remind me of something being like this, right? So, for example, Turing mechanism tells you that when you have large enough space, you go for what do you call this? Spots. And when you have rather cylindrical region, then you go for stripes. Okay, so usually tails have stripes and the body parts have spots, but there are exceptions. There are exceptions. So I presume that the patterns on giraffe is just the patterns that we see everywhere. Like even when the ground was wet for a while, then it becomes dry. It cracks, right? It cracks like hexagons. So I think that's what we see. So you see this is a general concept and it can produce all kinds of different patterns. So it describes giraffes' patterns, but the how part, not the why part. For giraffes, I think they are just, maybe if there are lots of giraffes that live together, do they?
As far as I know, yes.
Yes, so then yes. So then it works both ways because they also look like sand, right? So, but they are incredibly tall, so that's a problem. That's a problem. Very nice. Are we happy so far? So, what we have just picked in a little bit is morphogenesis, how shapes form in biology, right? Yeah, during morphogenesis, you have some signaling, right? Cells talk to each other and they go. So, you are incredibly complex human being, but you started with a single cell, right? That must be amazing. So let me show you a code that does nothing nearly as amazing, but something interesting. Ready? I will explain the code as well while it runs. So let me put it here, put this one here. Let me run it again. So you start with a single cell. It decides to divide, and it keeps dividing. It is signaling with other cells, and the middle one will die on its own. Okay, this is interesting. Very nice. Middle one should die. Yes. So this is a code that I specifically designed so that you start with a single cell, but you always end up with this ultimate shape. Do you see the ultimate shape? So even if you cut it, just like those things that have limbs regrowing, those animals, imagine that this is the shape of the animal, and no matter what, the system goes there. Okay? Now, how does this work? Well, they keep sending signals to each other, and they are also receiving those signals and deciding on their own fate based on those signals. And this is very accurate biologically. Like, that's exactly what also cells do. We have already seen, okay, I have a solution for that. Did you see the other state that is a local minimum, but it doesn't stay there? So that's by some tuning of the parameters so that it goes still here. All right. So basically, when it's alone, it sends some signals but receives none, right? In that time, it's just like leaky integration. Do you know what leaky integration is? Like you put something you have a pot, but it's leaky. You keep putting things in it. So it smells, it smells and it doesn't get any smell. It says, okay, I'm increasing probability of division. So if it doesn't receive a signal for a while, it decides that, okay, now I should divide. Then when it's 2, they still don't get enough signals from each other, so they divide again, and then they, at some point, these colors correspond to particular amount of signal coming from others, so they understand where they are. Okay? Yeah, this is what your body does. Do you know apoptosis? It's programmed death. Okay? Then there is necrosis. Necrosis is when it's unprogrammed, and when it's unprogrammed, then it's a mess. It's bad for your body. But if it's apoptosis, it's a plant death, so it doesn't happen like an explosion where you can't reuse the materials. What is your guess for how many cells kill themselves every day in a human adult? Just a guess. Trillion is the total sum. So how many in a million, right? That would be my guess as well. Turns out to be 70 billion, I think. Well, am I wrong? I'm still surprised with that word, so we can check it. Emin will check it. So, billions of cells are programming themselves to death so that everything runs smoothly. And that also happens with your hands, like you start like a duck, then these cells decide to die so that you can have fingers. Okay? Very good.
50 to 70 for adults. And
Billion, right?
Yeah.
Yeah.
And 20 to 30 was between 8 and 14 years old.
Very nice. Very nice. All right. So, speaking of going from single cell to fully fledged organism, we are used to it. We are used to everything being normal, right? We are surprised when a child is born still, right? Now, what is the mechanism? We should be surprised for it to be disorderly. That was bothering Stuart Kaufman. So, let me show you. Stuart Kaufman is here. Do you see? So, Stuart Kaufman wrote a book named Origins of Order. So he was wondering where the order comes from. And one of his main contribution to this field is he figured out where that order comes from, probably. So he came up with this Boolean genetic networks. I wouldn't say he came up with them. He understood how they work. So let's say I have, do you know how I can show my mouse while presenting?
Right click and laser pointer when you're full screen.
Do you see it here?
First, you need to start the presentation.
Yes.
Then right click and laser.
No, that's probably for. No, it doesn't work like that in Mac. Show pointer in Keynote. Settings, click slideshow. Show settings. Slideshow. Where is it?
It's loaded.
Ortalıcı. Aşağıda.
Second line.
Aynen.
Okay.
Tamam,
Thank you very much. Okay, here it is. So, are you still with me? This is epigenetics. So you have genes. Epigenetics is especially relevant during the development, okay? So it is used as a signaling. What am I going to do is, because genes will produce the relevant proteins will produce the relevant behavior, right? The relevant actions. So let's say you have 4 genes, okay? Epigenetics tells you that you can switch on or off the genes, okay? Now, imagine that every 2 gene is controlling a 3rd gene. The state of, so let's say the first and second genes are both on, then they send a signal that would switch on the 3rd gene. Does it make sense? Or switch off. We don't know. Neither did Kaufman. He just thought that let every 2 gene control 1 gene and let them have random rules. Okay? So, for example, we can have something like this. 1, 2, let's say we start here. So let's say everything is 0, 0, 0. So 0, 0 in 1, 2, 0, 0 turns on a 3rd gene, right? So that one becomes 1. Okay? Why this one is 0? Because 2 and 3 is controlling 4. Who is controlling 1? 3 and 4. 0, 0, 0, 0, 0. Do you see it? Everyone is seeing this? Yes? Now, then he puts it on a network, and he puts all the possibilities. For 4 genes, if you think binary, there are 16 possibilities. And if you plot it as a network, you can see that there is only one solution to this network. No matter where you start, if you start here, you end up being in this cycle. If you start here, you end up being in that cycle, etc. So far, so good? The thing is that you have 16 possibilities, but you have only one attractor, okay? Once he did it for much larger networks, he realized that if you have, let's say, 100 genes, and if you make a map, you end up with only 10 attractors. So far, did you follow this? He says that it's on the order of square root of the number of genes, the attractors. So he says that then probably these attractors are cell types. So during the development, you start with pluripotent cells or stem cells. They can become anything. Then while this signaling business is going on, like switching on, off the genes, in the end they emerge at one of those attractors, and if they end up here, then it becomes a neuron cell, end up here, becomes a kidney cell, etc. Did you follow this? So he says that then the number of cell types should be square root of number of genes. Or at least, when you plot it in log, cell types and number of genes should follow a line. Okay? Well, he does that. And, okay, let me give it. Everything in log is sitting on a line, but this is nice. Do you see? Like there's a man here, there's a jellyfish, bacteria, and number of genes and number of cell types are following similar patterns. So that's the origins of order. You can have 2 to the 100 states, but there is a very powerful attractor that attracts it, just like in the simulation that I have shown you. It always goes to the same kind of a structure, no matter what, despite the fact that there are some fluctuations, etc. Am I making sense? Okay. I think we can have a break if you don't have a question. Do you have a question? Okay, let's have a break.